دكتورة مروة حميد الخفاجي، دكتوراه في الأحياء المجهرية، مقرر التسمم الغذائي لطلبة الدكتوراه، علوم الحياة، كلية العلوم، جامعة بغداد. أسعد الله أوقاتكم بكل خير أحبتي، والسلام عليكم ورحمة الله وبركاته. Food poisoning and foodborne diseases. Food poisoning is an acute enteritis caused by the ingestion of food characterized by diarrhea, vomiting, with or without fever, abdominal pains. Food poisoning is normally associated with the small and large intestine. Certain types of food poisoning are described as intoxications, other as infections, and toxico-infections. We can define the foodborne diseases also as gastrointestinal disturbances resulting from the ingestion of food contaminated by any agent, chemical, biological, have a variety of causes, just like overeating, allergies, nutritional deficiencies, actual poisoning by chemicals, toxic plant, toxic animal, toxins produced by bacteria, infestation by animal parasites, and infection by microorganisms. As a historic preview, fatal diseases from eating blood sausage was recognized as early as 980. 1849, cholera through contaminated drinking water. 1870, the theory of potassium poisoning to explain illness contracted by eating certain foods. 1906, Bacillus cereus food poisoning was recognized. 1926, the first report of food poisoning by streptococci. 1937, paralytic shellfish poisoning was recognized. 1945, it is approved that the etiological status of Clostridium perfringens in food poisoning. 1951, Vibrio parahemolyticus was shown to be an agent of food poisoning, and the scomproid histamine associated poisoning was recognized. 1960, the production of aflatoxin by Aspergillus flavus was first reported. 1965, foodborne giardiasis was recognized. There are different types of intoxication. A. Intoxication caused by chemical substances such as drugs, pesticides, heavy metals, gases, solvents, where the patient has direct contact with the toxic substance. And B. Food poisoning of which transmission vehicle is the contaminated food with pathogens or chemical products. So either direct contact with chemical substance, or the contaminated food is the transmission vehicle, either with contact with pathogen or chemical product. Food poisoning or foodborne disease, FBD, is one of the main problems in public health worldwide. According to the WHO, World Health Organization, each year 600 million people around the world, or 1 out of 10, become ill after consuming contaminated food. Sources of foodborne pathogens. Many foodborne pathogens are ubiquitous in nature and generally found in soil, water, animals, plants. Pathogens are introduced to processing a plant through the raw materials. Human and plant equipment are may also bring the organism to the food plant. This table shows the persistence of foodborne pathogens on inanimate surfaces. So these are the organisms or the microorganisms, and the second column about the duration of survival on inanimate surfaces. Just like Campylobacter jejuni, up to 6 days on inanimate surface. Listeria, 24 hours to several months. Norovirus, 8 hours to 7 days. Hepatitis A virus, 2 hours to 60 days. So it is different according to the species and circumstances. There are many types of food poisoning, but we can estimate 3 initial types: food intoxication, food infection, and food toxico-infection. In the first type, food intoxication, a foodborne illness occurs as a consequence of ingesting a pre-produced bacterial or fungal toxins or toxic secondary metabolites during their growth in a food. So, the intoxication is a foodborne illness occurs as a consequence of ingesting pre-produced bacterial or fungal toxins or toxic secondary metabolites during their growth in a food. The toxic metabolites present in the food and not the viable cells are responsible for such virulence. So we ingest contaminated food with toxins, not the viable cells. After the production of toxins, there is no need to living cells to be present in food for further illness. Just we ingest the contaminated food with toxins. There is no need for living cells to produce such intoxication. The second type is food infection. This foodborne disease or illness occurs after the consumption of contaminated food or water that encompasses live enteropathogenic bacteria or virus. So necessarily viable cells of microbes remain alive in the food or water during consumption. Being present in small numbers, these pathogens have the potential to establish and multiply in the host and reaction of the body to their presence or their metabolites to cause this illness. So we must ingest contaminated food with live bacteria or virus. Even in small numbers, these pathogens have the potential to establish and multiply in the host, and the hosts react to their presence or their metabolites. This is the food infection. The third type is toxico-infection. This type of illness may occur due to ingestion of bulk of viable pathogen through contaminated food and water in the due course of infection. Consequently, the microbes either sporulate or die, releasing toxin to stabilize further disease manifestation with related symptoms. So we must ingest food contaminated with viable pathogens in high numbers, so either sporulate in our body or die, releasing toxin to cause such type of food poisoning, the toxico-infection. consequence or sequence of events in a food poisoning. Initially, there has to be a source of pathogen. Next, the pathogen has to contaminate food. Consumption of the food. Consumption of the food contaminated with this pathogen or pathogenic virus can lead to viral infection. For bacterial pathogens and toxicogenic mold, the contaminated food has support the growth and to be exposed for a certain period of time at a suitable temperature to enable the pathogen to grow. However, for some potent pathogens such as Escherichia coli or 157H7, growth may not be necessary to cause foodborne infection. It is a potent pathogen. For intoxication, growth should reach a sufficient level to produce enough toxin so that when the food is consumed, the individual develops the symptoms. For bacterial infection, viable cells of the pathogen need to be consumed in sufficient number, which vary greatly with pathogens, to survive stomach acidity and establish in the digestive tract and cause illness. For toxico-infection, viable cells should be consumed either in high numbers for those cannot multiply in digestive tract, such as Clostridium perfringens, or in reasonable numbers for those can multiply in digestive tract, such as Vibrio cholerae, so that toxins released by them in the digestive tract can produce the symptoms. In this diagram, the sequence events leading to a food poisoning by pathogenic bacteria and virus. Here we see the reservoir of a pathogen, then the contaminated food, either contaminated by a virus, so it is viral infection, occur when we consume the food. When we consume the food. Long storage of food at a growth temperature lead to growth of pathogens to sufficient level, causing one of the three types of food poisoning: intoxication, infection, or toxico-infection. The intoxication occurs after the consumption of food containing toxin. There is no need to live cells. The infection occurs when we consume food containing live cells. Toxico-infection occurs when we consume food containing live cells in sufficient numbers. We may hear about epidemiology. Epidemiology has been defined as the study of how often diseases occur in different groups of people, and why epidemiologists are in many ways disease detectives, tracking down diseases to discover why it is occur as well as when and where it occurs. Epidemiology of food poisoning. Many individuals may be affected by food from a single source. Family food poisoning occurs when some or all the members of a single family exhibit symptoms of food poisoning. Sporadic cases involve individuals only. Epidemic incidents may be explosive if a large number of people in a single institution or attending the same function are simultaneously affected. So we have family food poisoning, sporadic, and epidemic incidents. Alternatively, cases may be dispersed of food from a point of source from a point source is transported over a wide area. So we have dispersed cases in this situation. The easiest incidents to study are epidemics, and the most difficult to examine are sporadic incidents, because the high incidence of sporadic cases, it is very difficult to assess the true scale and nature of the food poisoning. This problem is exacerbated by the fact that in many cases of food poisonings are a self-limiting condition and relatively short period. Global epidemiology, food poisoning and foodborne illness is an important cause of morbidity and mortality worldwide. In 2015, the World Health Organization published its first estimate of a global foodborne disease incidence, mortality, and disease burden in terms of disability adjusted life years. Disability adjusted life years, DALYs, caused by 31 agents including bacteria, viruses, parasites, and chemicals. The global burden of foodborne diseases caused was estimated to be 33 million disability adjusted life years. The epidemiological triad. At the top, agent, causative factors, risk factors, environmental exposures. The host, person characteristics, group and population demographics. Environment, place characteristics, biological, physical, and psychosocial environments. Time, we have time characteristics, incubation and latency, length of disease process, trends and cycles. We have two important terms in the epidemiology: morbidity and mortality. Morbidity, life. Mortality, death. Morbidity, proportion of illness in a population. Proportion of illness in a population. While mortality, incidence of deaths in a population. Incidence of deaths in a population. So morbidity, live people or population, mortality, death, population. Morbidity, proportion of illness in a population. They are still alive. While mortality, incidence of deaths in a population. We see in this slide animal origin food and there being a reservoir for many pathogenic agents causing epidemiologic diseases, just like Creutzfeldt-Jakob disease, E. coli, Creutzfeldt-Jakob disease with cow, E. coli with sheep, Campylobacter with chicken, Salmonella with chicks, avian flu with birds, brucellosis with goat, swine flu H1N1, with pigs, SARS with bat, in addition to COVID-19, also from bat. Thank you so much for your listening.